Hi everyone, I'm Melanie Ferlazzo and I'm a postdoc at the Australian Nuclear Science and Technology Organization. Today's topic will be human radiobiology in space. But before taking you to space, a bit of background on this project. This is a collaboration between France and Australia. On the French side, you have the French Space Agency and INSERM U1296, which is my former lab. And on the Australian side, you'll have collaboration of the Australian Space Agency, ANU, UTS for a project linking radiation and microgravity, ANSI for some funding, and of course, ANSTO. Individual radiosensitivity is not a new idea. This is something that was discovered about 100 years ago, not too far from the discovery of radiation. And the main key point is that we are not equal faced to radiation. This is a forgotten evidence, and it's true for both high and low dose. It's a legal, ethical, and societal issue. In 2019, my former lab participated to the writing of a French new decree about this individual radiosensitivity. So what is it exactly and how does it show? The main way to observe individual radiosensitivity is to go to radiation therapy service. You will see people that after their radiotherapy treatment will show skin burns. This is a physiological manifestation of a cellular problem. So what we do since 2003 with the Copernic project is that we collect skin biopsy from about 30 hospitals in France, some in Belgium too. And from that skin biopsy, we do cell culture and then we irradiate the cells with the same machine that the patient will go through. We're looking at DNA repair and recognition mechanism. And with an algorithm and a radio sensitivity diagnosis, will help oncologists to adjust the treatment for the patient and decrease the number of secondary reaction. So what is our hypothesis? ATM, which stands for ataxia-telangiectasia mutated, is a protein that will shuttle from the cytoplasm to the nucleus in order to recognize double-strand break and trigger their repair. That nucleoshutting was found systematically delayed in the radiosensitive patient. It is observed by immunofluorescence, Western blot, and mass spectrometry. How does it work? So we'll separate the population in three groups. Group 1 is your control population, the normal populations, about 75 to 85% of the individuals. In normal condition, ATM is in the form of dimer in the cytoplasm, and after radiation, due to the oxidation of the environment, those dimer will separate to monomers and then go to the nucleus to recognize and initiate DNA repair. At the other extremity of the spectrum, you have group 3. Group 3 are people with diseases that will impair the DNA recognition and repair pathway proteins. One of the examples that is the most common and gives you the bigger radiosensitivity is the ataxia-telangiectasia disease, where ATM is mutated. So even if it separates to monomers and go to the nuclei, it doesn't change anything because the protein is not active. In between those two extremes, you have group 2, which is about 5 to 20% of individuals. These individuals are the patient that will show secondary reaction after the radiotherapy treatment. The ATM nucleoshutting is delayed, the DSB recognition is incomplete, the DSB repair is incomplete, and they show moderate radiosensitivity, but significant one. They also have a high risk of cancer and a high risk of cancer after radiation therapy. During my PhD, I identified X proteins that are shown to interact with ATM and create that delay. We'll see a bit later how important these discoveries are in terms of the exploration of new radioprotective solution. So following the radio-induced ATM nucleoshuttling, we can identify three major consequences of an irradiation. You will have the radiosensitivity, which is the tissue reaction I just mentioned, radiosusceptibility, which is a transformation in the cancer prognosis, and also radiogeneration. radiodegeneration, which is senescence and aging. What we need to do now is try to take that ATM nucleoshutting model and apply it to find a novel radioprotective approach. The usual antioxidant agent will try to reduce the number of double-strand break induced. What we want to do here with the ATM nucleoshutting model is try to accelerate it in order to improve the radiation response. So this is what we do with the Zopra treatment. Zopra treatment is a combination of zoledronate plus pravastatin, and it's been shown that if you put that on your cells before radiation, those X proteins that are linked to ATM will separate and allow the nucleoshuttling to happen. So all those proteins that I've identified previously, we've applied the treatment and we can reinstore a normal response to radiation to those cells. What we can do also is try to use different types of radiation. So the results I've showed you earlier are made with gamma rays. But as you can see, if you radiate with carbon ion, you will have a problem of recognition and repair of 24 hours of radiation. If you put the Zopra treatment to those cells that are radiate with carbon ions, you increase significantly the recognition of the breaks, and you also decrease the number of breaks not repaired after 24 hours. It's not perfect, but it's a nice improvement. So one of the ATM nucleoshuttling theory application could be done for space and the development of new radioprotective solution. So speaking about space, space is a big radiobiology challenge. You have different types of radiation. So the main component are the galactic and solar radiation. That is mainly composed of protons, some alpha particles, and some heavy nucleus. You also, so here on Earth, we are protected by the Van Allen belt, and we have a natural background radiation, which for Australia is about 2.2 millisievert per year. In Iran, you have 70 millisievert per year. That's the highest background radiation that you can find on the planet. And you can see that even that higher radiation background is still twice as less as what you can find on ISS. So if you spend a year in ISS, you will have double that background radiation, and hence the need to study the effect of such an exposure. And if you go to Moon or Mars, you'll increase that background radiation by about 2. And if we imagine a mission to Mars, you need to take into account the time travel that you will spend to go there. So for a 2 to 3 year mission, it's estimated that the astronaut will receive 1 to 1.3 millisievert per year. So that kind of exposure needs to be studied before sending people. It's also a combined effect of radiation. So you will have those proton, those alpha particle, that background radiation, plus a secondary radiation, which is the result of the interaction of the primary galactic mix with the shielding of your spacecraft. So those radiation need also to be studied. And this project will study the DNA recognition and repair mechanism and also the mitochondrial activity. We have a project with UTS to investigate the effect of radiation combined with microgravity, and we also aim to offer some countermeasures. So what's our approach? We are interested by 2 main components of the spatial radiation mix: the secondary low energy particle and metallic ion that are generated from the shielding, and a constant low dose bath of radiation, about 0.4 millisievert per day. And we will use 2 different types of tissue: surface tissue for the low energy particles, such as skin and eyes. So I've started the experiment with skin at the moment. and will, back in Lyon, some people studying the eyes, and also deeper tissue for that bath of low dose of radiation, such as bones, heart, and brain. Space radiation exposure for the low dose part is an equivalent to the low doses that you are exposed to radiodiagnostics. From that graph that sum up all the mission and their relative dose, you can say that those 0.2 to 0.4 milligram per mission days are the equivalent of one mammography view every 10 to 20 days or one CT scan every 50 to 100 days. So that's twice the highest natural radiation background. For the secondary radiation, here I will simplify doodle that explains how it works. You will have the primary particle from cosmic and galactic that react with your shielding and create metallic particle and low energy protons. What you see here is an helmet from an Apollo mission. So this helmet didn't go outside, didn't go for extravehicular activity, but when they came back, they analyzed the helmet, and you can see tracks. There are actually tracks made by a metallic particle or proton. So there's something that goes through the shielding, and it needs to be investigated. And that's clearly why I'm here at ANSTO with the new external enclosed ion beam, which is the first dedicated electronic testing ion beam facility in Australia. We will study the effect of those low energy proton and metallic particle. So what we had to do, we had to upgrade the entire beamline to extract the beam from vacuum to ambient air, because you can't really work with the cells on vacuum. You need to be in normal atmospheric pressure. So with this beam, we'll work on cells. That's my main project. But we also had space infrastructure funding project from the Australian Space Agency, which is a collaborative project with ANU, University of Wollongong, CMRP, Australian Synchrotron, Sabre, Nova, and Ceratec. And this will use a facility as a payload testing for a space mission. We also plan on using microdosimetry to have a cellular level dosimetry in collaboration with University of Wollongong. So now we'll take a closer look of the beamline. So here you have the external chamber on that pictures, and here are my it's a little cell holder that we've been working on. And actually what I'm doing is that I put my cell in that tiny membrane here. So this is 75 nanometer thick membrane, so we can be as close as we can to the beam. And so the first experiment that happened right before lockdown was just a simple proof of concept and proof of feasibility. Here you get my human cells that I've seeded on that tiny membrane, and we did 2 simple irradiation, and then use our immunocytochemistry to have a look at DNA breaks. And yes, here we are. We can see the double strand break after radiation on these cells on that very tiny membrane. So the idea now is that the radio-induced ATM nucleoshutting model, it's a general mechanistic model that study and predict the individual response to radiation, of course. It has application in space radiobiology to better understand the response to different types of radiation, and to also create novel radioprotective strategy. We consider space radiation in the spacecraft through the radiosensitivity, radiosusceptibility, and the radiodegeneration notion, and also the effect on the mitochondrial pathway, because ANSTO is quite good at characterizing the mitochondrial states. So what's next? With that high energy heavy ion microprobe, of course, when we back to the lab, we'll keep going on the DNA analysis. We'll change the cell type, use different genetic background, and we'll module the dose in the particles. We'll also study the mitochondrial activity, protein analysis, do RNA analysis, reactive oxygen species analysis, and here again, we'll different dose, different types of particles. The beamline can also accommodate different types of living sample. So we could also study the microbiome, the effect of those types of radiation on food, on nutrition, on drugs. We can also try to find a solution for the problem of biofilm that is created in the different spacecraft, and we can also study plants, of course. The beamline is open to collaboration. It's a great tool for research and industry. I would like to thank everyone involved in that project. Of course, that wouldn't be possible without, of course, my former lab in Lyon and the great team at ANSTO, Human Health Bioscience CAS, and also like to thank all the sponsored and all the collaborators that we have here with that project. Thank you very much.